And thanks for the invitation. This is a great opportunity to tell you a little bit about some of the wondrous materials that nature makes. Along the process, if I don't speak loud enough, I want you to yell. And I'm pretty informal. And if you have a question in the middle of the process or I talk too fast, you should yell out your question as well, and I'm happy to stop. We can make this as informal as we'd like. So let's see if this works. Oh, yeah. All right. So just a brief structure of the type of materials and the types of topics we're going to talk about. Very briefly, at first, I'm just going to give you a feel for some science-made materials that we make in laboratories all over the world, all right? And the objective here, and all I want you to take from this initial slides, is for you to understand what types of materials are made, how intricate those materials are, and the types of instruments that they use to make those materials. And we're going to contrast that to nature-made materials. And we're going to look at the organisms that make those materials, the techniques that those organisms use to make those materials, and the function of those materials in the organism. And then later, I'm going to tell you, hopefully we have time, explain what some different types of scientists do. Because while I think you guys are a lot more sophisticated than I was in high school, I didn't really have a good feel for what scientists actually did beyond the chemists that I had classes from and the biologists. So I hope to give you a feel for that. Okay, so before we get started on the types of materials that we're going to look at, I want to give us an explanation of the scale of the materials we're working at. So my ruler in this case is a little different than our normal rulers we have in our desks. So my ruler starts at 0.1 nanometer and goes to 100 micrometers, 100 microns. So if you touch one of your hairs and you can manage to just get one hair and you feel the width of that hair is around 60 to 120 micrometers wide. So it's very tiny, so you can barely feel that one hair. And you look at other things that are about that same size, that's like a dust mite. If you live in a humid environment like California, dust mite is about the same size. You go smaller, this is a picture of a cell. So human cells and plant cells are around 10 to 100 micrometers. You go even smaller, and now you get to the viruses. These are about 10 to 100 nanometers also, much smaller. That's why they can get into little places that are much harder for bacteria and other things to get into. These are called organelles. These are part of the cellular machinery of these cells, and they're obviously about 100 nanometers as well. And as you get smaller than the viruses, again, you get to individual proteins, which we're going to talk about in a second. You see carbon nanotubes, which you may have heard about in the news. And then eventually, when you get really small, you start to go into the molecular regime. So our first man-made material, the architects are two colleagues of mine, Amit and Mike. The interesting things about these materials is the first thing, what they're able to do is they use this really cool instrument called an electron beam evaporator. And in that electron beam evaporator, they're able to put down layers of different types of metals. So in this example, they put down 2 nanometers of copper. Then on top of that, they put down 2 nanometers of niobium, and they keep alternating this. This is what they constantly do. Now, that's pretty cool because the structure is really tiny, right? And they're able to do this with a huge amount of precision. But the really neat thing, and this is the only plot I'm going to show you, is that when you make these materials in the nanoscale and you really control their architecture, you make them incredibly strong. And so these are the normal materials that we have in planes, trains, and automobiles. These are made by metallurgic processes on the left. You can look at their strength. It's not nearly as high as these nanomaterials, which are about 1,000 times stronger, okay? So very intricate, very strong, and made with these huge instruments. All right, the next materials architect is a colleague of mine, Jen Hollingsworth. Jen and her group and a number of other scientists at the lab make what are called quantum dots. And these are basically just collections of metals. And when you get these collections of metals close to each other, they fluoresce. They're really bright, and that's what you see here. So if you look at this electron micrograph, they can also make these rods, and they make these rods with really nice uniformity. And this is a picture of a cell that's being lit up by these quantum dots. You can see the nucleus as well as the other parts of the cell also. But what I want you to take from this is how, as synthetic chemists, we make these types of materials very high temperature, 360 degrees Celsius, okay? And organic solvents, so things that are not necessarily very green chemistry type stuff. And we often use these things called glove boxes so that we keep us away from the material and the material away from oxygen. But amazing precision. Oop, still figuring this thing out. All right, the next example, the architects are a friend of mine, John O'Hara and Rick Averitt, and then these guys are all bunnied up in this bunny suit. They work inside of what's called a clean room, and the idea here is that these guys are able to make, in this clean room, these really intricate structures that you can only make under these very purified conditions. But the cool thing is if you look at this middle picture, you're able to make structures that are kind of like the cogs of a bicycle wheel, but instead of being this big and running into each other, these things are like 10 micrometers, so even smaller than that grain of your hair. And they can turn and they can move, but they can only make them if they can suit up and go into this large machinery. So let's look again at our ruler, the same scale. We see all the different sizes of these materials that we just talked about. What do we know? We know that we can make these materials on the science bench with amazing precision. These are at the cutting edge of making these types of materials. The materials have tremendous properties and gives them great applications. And they're made maybe in minutes, hours to weeks. It takes a while to characterize these. So it takes a while to make good materials. And we know that they're instrument intensive. So you have to have big instruments, solvents, high temperature, and vacuum to make these well. So let's look at how nature makes materials, and let's look at some of the materials that nature makes. So nature, and I hope you'll be able to see this from these pictures, nature makes materials that are very, very precise. Just like the same precision you saw in the man-made materials, nature can do the same thing. They're small materials, but they're really strong. Nature makes materials in minutes to hours, not necessarily days or weeks. It's fast. The other really amazing thing about nature is that it's programmed. Inside of that cell is the DNA that's specific for that cell, and that DNA tells this cell how to make that material step by step by step. And every cell has different information, and that different information makes a different material. And because it's programmed, it's very, very reproducible. And that is something that is hard for us to do synthetically, to make things over and over the same way. And cells are able to do this. So this leads to a field called biomimetics. And I'm going to show you some of the biomimetics at the end. But one of the objectives is to use biology and to understand from biology so that we can make materials that are as good as nature can make materials. We want to make them fast, cheap, and reproducibly. And that's what nature is really good at. So I'm going to show you three different types of materials that come from three very different types of organisms, okay? The first one is called a diatom. Diatoms are unicellular organisms. They're eukaryotes. They're photosynthetic. That basically means they take the solar energy from the sun, and they take carbon dioxide, and they make it into sugar. So that's what they live on, is that sugar, and they transport that sugar to other organisms. So you may have been hearing talks about global energy and global climate change, and they talk about the problem that we're making a lot of CO2, and they don't really know what to do with it. Well, it turns out when you look naturally, who changes that CO2 into sugars, which is what we need for our environment, turns out that anywhere from 25 to 40% of that CO2 gets eaten by these diatoms. So they're doing a really good thing for us in the Earth. And so while I like them because they do this great thing, it turns out they're an amazing material. And so when you look at that diatom, the diatom itself is kind of a squishy, soft material. But when you look at the diatom itself, it's protected from the ocean by making what's called a frustral. And a frustral, you're going to see, is kind of like a shoebox with two exact symmetric sides. And this frustral fits together with a girdle that holds these two shoebox sides together. And what I'm going to show you, and I want you to appreciate as you go through these slides, is that shoebox is made of glass. So this diatom is able to make these really intricate structures out of glass. So this glass has a really high strength to weight ratio. Not only are these diatom frustrals amazing materials, but I think they're just plain beautiful. And so that's what we're going to look at. So let's look at a few different types of diatoms. So these are black and whites of different diatoms. What I want you to see is that each diatom looks different, right? This guy, I think, is kind of like a crown, an eyeball-looking thing. Who knows what that is? Earth, and all these odd shapes. The reason that they all look different is because each one of these is a different type of diatom. And every time that this crown here replicates, its children all have that same frustral that looks exactly like this. And that's what's pretty amazing about these. So let's look at a few more. These are all different types of diatoms. You can see they all look different. The other thing to notice is how symmetric these diatoms are, right? This has a three-dimensional symmetry. And so not only do they have amazing precision at this 100 micrometer scale, but when you zoom in to just this little spot and you zoom down to here, you see that that symmetry is, carries down to a much smaller scale. Okay, so this is like a whole bunch of sieves, and these are even smaller sieves.
So could you tell us a little bit about how you make one of these?
I'm going to tell you. Yeah, exactly. That's the curious thing, and this is what people have been trying to figure out for a long many years. Yeah, so then this one's actually not colored in reality. This is just a fancy artist rendition of electron micrograph. Again, a few more different looks. This is again a blow-up of some of these sieves, kind of like the same materials that you use in the kitchen to drain your grapes or spinach or something like that, but these are on the nanometer scale. Again, here, these always look like microphones or headphones to me. And this guy, I think, I just call him the condo of diatoms. I don't really know what's going on with that one. These are different girdles in the diatom. So I told you those frustrals are held together with a material called a girdle. And what you can see is that these girdles have teeth. So every time this diatom replicates, it makes these teeth that are only like 5 nanometers, and those teeth stick together. And every time it makes those teeth, which are just tiny. Far, far smaller than the size of your hair. So if we look at the scale, we can see that diatoms range in scale size. Some of them are only a micron. Some are 10. Some are hundreds of microns. They all look different. And when we zoom into them, they all have different substructures that we can see that are really intricate. So how do they make them? Well, this has been a long, many years of discovery. And I said it's sort of like materials forensics, because it turns out that now we have the capability of looking at the DNA, so we can sequence the genome. But along many years ago, they didn't have that ability. And so they had to do a lot of materials forensics to try to understand how these are made. So one of the first things they noticed is that frustral, that glass house that the diatom lives in, is not just silica. It turns out there's a lot of peptides and proteins. So protein kind of like the protein that you have in your eggs, right? So that frustral is covered with peptides and protein. Not only is it covered with those peptides and proteins, but if you look and you bore inside of that silica, there's actually peptides and proteins inside the silica as well. They also noticed that each diatom has that different structural pattern, which is what we saw earlier. And they hypothesized early that that meant that each diatom had different peptides and proteins. And now, after years of being able to do research, they realized they were right. Very amazing things about this is that the formation of this nanostructured silica, so the formation of that glass house, is under genetic control. That diatom has the DNA that tells it how to make that material. And the cool thing is you make these really intricate glass houses that you saw out of shapeless starting material. So how do we do it? So this is a lot of stuff on this slide, so let's just go through it step by step. So number one, you have to form a reaction vessel. Okay? So when this diatom splits and you have 2 daughter cells, each cell has to come away with a frustral, otherwise it'd be damaged, right? So what it does is when it's splitting, the cell is covered by a lipid membrane, which is just kind of like an oily globular material. And that oily globular material actually makes a balloon inside of itself. And that's what's sort of shown here. I called this the silica deposition balloon here. So it needs to make a reaction vessel. So it makes that out of its own cell membranes. It steals itself. It steals it from its own self. The second thing it has to do in this section is it has to get its starting materials for glass into the diatom. Those starting materials are silicic acid, and there's a lot of that in the ocean, but it doesn't do them any good if it stays in the ocean. It actually has to come into this diatom. And so to do this, the diatom has these things called silica transporters. And these are just proteins that sit on that membrane and move silica from the ocean into the organism and keep pumping it in. It's just a constant pump, like a well pump. Now, they have to get inside the diatom, but they also need to get inside of this balloon. And no one's entirely certain how that happens, but they know it happens, and if it doesn't have these pumps, it doesn't get inside. Well, now here's the quandary. So now you've taken a ton of this silicic acid, and you've put it inside of the diatom, and now it's at a very, very high concentration. And if you leave it in that diatom by itself, it'll actually polymerize and start to make glass all on itself. And the problem with that is that if it just makes a random amount of glass, you don't have those pretty structures that we saw in the diatom. You have this amorphous gunk, kind of like accidentally making jello in all these different forms. And that would probably kill the cell. So the diatom's smarter than that, and in its DNA, it has the genetic information to tell proteins to make what are called polyamines. And these polyamines are positively charged, and what it does is basically wrap around that negatively charged starting material, the silicic acid, and it stabilizes it, and it keeps it from getting too close together so that it doesn't self-polymerize. Then you have to make the pattern. That's number 3, right? Because we saw that each of these diatoms makes a very, very intricate pattern. So next you have to make a pattern. Well, it turns out that when they started to sequence these genomes and they looked at the different types of proteins that are in them, they see that each diatom has a set of proteins called silifins and silicidines and frustulins and pluralins and all these names. They basically just takes the word silica and add some names to it. And they figure out the structures of these. And it turns out what they've found is that when you mix these silicidines and these silifins with these guys, they actually cause a phase separation. So, you know, if you've ever taken wet lettuce and you accidentally put some oil on it, or you're going to make a salad, and you look at what happens on that wet lettuce, it makes drops of oil, right? It doesn't actually mix with the water on the lettuce. Well, that's the same thing that's happening. These silifins, or these proteins here, peptides, aren't actually mixing with the polyamines very well. And so what happens is that oil and water separates. And that's what's sort of shown here. So if the white dots you can think of are the silifins, and the black is silica, okay, and this polyamine. And so these proteins are actually sort of the negative, kind of the bricks, and the mortar in this case is the silica. And so that's what forms the pattern that you saw that's highly intricate. There's also a group of people that think that the bigger structures that you saw are because the cell actually sacrifices part of itself to make this frustral. And what it's sacrificing in this case are its own organelles, its energy machines. And these energy machines get lined up in particular shapes and orientations because it has that genetic information to tell it to do that. And then you put silica all around it as well. So the same idea. So the important point to take from this is that you're able to make, it's genetically controlled, and that you're able to make these very hard materials that are very intricate just out of a soluble precursor that's abundant in the ocean, silicic acid. All right, so the next example is not a solution-based chemistry example. This is a physics-based example out of biology. So this is what I called structural color in butterflies and beetles. So our architect for these materials are over here. This is a weevil. I don't remember his name, but he's way cool. And if you zoom in on him, this is the back, one of these little spots that you see here. And what you see is that each scale is this really brightly colored yellow. And it's kind of iridescent. As you kind of look at it, it changes color, right? And then you have some green ones, and then you have some blue ones, and it cycles in, and you finally just get the black back of that beetle. So I'm going to give you a hint to how this works, and we're going to first look at some pictures of these weevils and butterflies. But I want to just start to give you a hint of how it is. This is something called a photonic structure. This is made by science. This is a laboratory-made material. It takes those guys in those bunny suits to be able to make this really intricate material. You can see that scale. It's really, really small. The cool thing about these materials, and this is all you have to understand from this, is that because of their structures, they change and control how light interacts with them. So when light hits that photonic structure, it bounces back off in a particular way. And if I change the spacing of those lines together, it'll bounce off another way. And so what I see, my perception is, it's a difference in color. So why would scientists be interested in this? Well, you know, our US dollars, our $5 and our $20 have finally gotten fancy, right? And now they have all these anti-counterfeit things. So when you hold them up to the sky, you can see all these really cool formations. Well, they'd like to make dollars and all these other things even fancier. And so they want to add some of these structured photonic materials in there so that they have these iridescent properties. Telecommunications are interested in them. They're being used in cosmetics and paint as well. So let's look back at the butterflies and beetles and see how they relate. So think about this color. So this color has to come from somewhere. It can come from pigments, which is, you know, the pigments in our clothes, or it can come from what is called structural color. And in fact, I'm going to tell you almost all of the color in these kind of things, especially also in some of the fancy fishes that Rubottom showed you guys a couple weeks ago or months ago. If you see something that's iridescent, it's coming most likely from structural color. So why structural color, and what is it? We're going to get back to that. But structural color gives you a very high intensity. It gives you a variety in color. It also is the reason that you see that iridescence, right? So like when you look at a peacock tail and you kind of change your head, the colors sort of change, right? And that's this iridescent or a pearl look to something. So what do they use this for, the peacocks and the butterflies and beetles? Well, mate attraction. Next is communication. They think it's also part of migration. It tells them how to move and to see their kins as they're moving along to the south. And it's part of camouflage. And we're going to see a couple of those little guys that use it for camouflage. So I'm going to bring this subject up again, but there's this really cool butterfly in the Amazon. I think his name's Morpho. And it's a big butterfly. And what they say is that if you're actually above all of the trees and you see Morpho flying, that you can see the light off of one of his wings a half a mile away. And the reason that you can see that color half a mile away is because that guy, not to ruin the story, has some really cool structures in it that amplify the light. So let's look at some cool pictures of butterflies. So I think this is Morpho, but I could have this wrong. I'm not a butterfly person, but this is my impression. You can see all the iridescence, and that, again, is the structural color. This guy, the iridescence is in the green. And here again is sort of a blue purplish iridescence. This is a moth. It's pretty cool. He's kind of a hairy moth. And on his back wings here, you see this purple and orange iridescence. These are scales of butterflies. And never until I started looking at this research did I realize butterflies actually have scales, but you're going to see that they're full of scales. And when they plucked off those scales and they looked at them under a microscope, they see that they're really iridescent like that. And this guy is the wing of another butterfly that he uses it for camouflage because he wants that eye-looking thing to appear like an eye of a big bird or something else, a predator. And when you move relative to that structure, because it's iridescent, it looks like that eye is moving. And that's because of the structural color. So butterflies are cool, but I think you're going to see, I hope, the beetles are even cooler, okay? So beetles have long been called the living jewels. All right, and beetles are really beetles have also been used, I think, as art for back in the old Egyptian times. They had made pins out of beetles for women to wear. You can see why these beetles are incredibly gorgeous. They have all these metallic sort of colors, and they have all these different iridescent colors. And when you blow up onto, you kind of look at an amplified view of their back, you can see that they have just these sort of pearlescent qualities about them. Some more beetles. These are pretty amazing little creatures. You can see that these look like gold. This guy's called the 24-karat bug because he looks like he's made of gold. But I guarantee you, if you grind him up, you're not going to find any gold. All you're going to find are optical and photonic structures. Okay, so you'll be very disappointed in these times when gold is selling high. This guy's not your fortune. But these are pretty amazing. This next one's my favorite. This guy makes these little dots that when you look at him close are actually these arrangements of hexagon-shaped materials that look like opals. That's basically what an opal looks like if you look at it very close. And so they're very highly iridescent. All right, so how do they do it? Just ignore this for now, all right? I'm just going to tell you that butterflies and beetles have 4 strategies, basically, to make structural color. One, the first one here, is they make multiple layers of material, and they control the spacing between those materials. So instead of just one layer of material that light can hit and bounce off, they have many, many layers, and they control how much air is between those layers. And the idea here is that when light hits those layers and bounces off, if they control that spacing, they don't just have one light wave going through that, and they can't just see that one light wave. They have multiple ones. So there's 2. There's 10 layers. Now you have a big signal. So they boost the signal that you see in that color. That's why when Morpho is flying a half a mile away, you can see the spark off of his wing. If it was pigment, you wouldn't be able to see that. But because it's structural color and Morpho has a lot of these different layers, you're able to get this huge amplification, like turning up the volume on your headphones. So they do that. They also make layers out of very different materials, as well as different thicknesses of materials. And that changes how light interacts with them. And then the cool thing, this is the 4th one, and I think you're going to see that beetles, they're so cool because not only do they look beautiful, but they actually incorporate the most interesting physics and most interesting materials. And they make what are called 3D photonic crystals. Bottom line is this, that these organisms interact with light by changing the type of materials that they have on their shell. Again, this is under genetic control. Yeah. Okay. All right, so how do they do it? Let's take a look first on the left. This is a cartoon view of a butterfly. So like I said, it turns out that butterfly wings are covered by scales, and there are actually multiple layers of these scales, and they sort of interdigitate. That's kind of what they're showing here. And if you zoom in on one of those scales, you actually see that the scale is made up of a lot of layers of material that are really controlled spacing, just like I said is one of the strategies. These are called cuticles. And when you zoom in again on that cuticle, you see that it's not just a planar, a flat surface of layers. Each of these cuticles is actually corrugated, like a corrugated box, and it goes like that. It's in a wave. And that changes how light interacts with it. So these are actual images. This is a cartoon. These are images of this butterfly wing. These are electron micrographs. These are different layers of cuticles. And it's a little hard to see maybe, but you can hopefully see that each one, there's like 7 different layers in there. And if you look carefully enough, you'll see that they're really controlled, the thicknesses and the layer and spacing between them. And when you zoom in fact, you see something just like that. You see that each cuticle is like a corrugated box. So they control light by having multiple layers so that you get this amplification. And they control light by the structures on these cuticles so that it interacts differently with the light and makes you see something different. These are just some more electron micrographs because I'm just amazed that we can see nature at this kind of magnification. So in my mind, this is perfect for the O'Keeffe kind of thing because it is art from science. These are the scales. This is already at 1,000 times magnification from what your eye can see. 1,000 times to be able to see this structure. That's why I had no idea that there are scales on butterflies. I can't see that far. This is 2,400 times scale. You can start to see those cuticles come out. And when you zoom in 25,000 times amplification from what you can see with your eyes, you can start to see that corrugation, right? You can see them sticking out here. This, on the right, is a biomimetic. Some scientists saw how cool these butterfly wings were and figured out how to make them in the lab. So nature is an inspiration to science. All right, but I told you, I think I have I like beetles better. So beetles have a really cool little ability also. So let's start on the bottom right. This is that's the beetle, or weevil, I think it is. The weevil, the back of one of those spots. Now, if you looked carefully enough, that beetle has sort of like little scales. That's what it looks like. They call them something else. And that's what that color is on. This is a magnification of that scale. It's kind of like a fingernail that it has a little tentacle and it's stuck hard. It's like a tack, I guess you can think of it. And so it sticks onto the back of that weevil, and when you accidentally take the scale off, it leaves these indentations. So now we're going to magnify that scale even further. So that's this next one. So how does the beetle get its color? Two things. It has the tricks of the butterfly. That's nothing to it. So it has these like 10 layers of a material, and those cause that amplification that we were talking about. But underneath it, if you drill even further, it has this photonic crystal. And this is a fairly new discovery. This one was discovered just a couple years ago. And now the physicists are starting to get really interested, because now they realize that this is an inspiration for a new type of material. Well, this is the other interesting thing. So what are these materials made out of? In this case, it's made out of a molecule called chitin. Chitin is actually just a sugar that is made by the weevil, and it polymerizes and forms a long chain of sugars. Well, the funny thing is that this structure up here is chitin, these layers, and these hexagons that form that photonic material are chitin also. But somehow the organism knows to take chitin and make long strands and pack them into layers of paint here, and yet it's able to figure out how to make chitin into a hexagon and to pack that differently here. And so I'll tell you, this is a pretty new discovery. They haven't been working on this nearly as long as they've been working on the diatom. So they don't really have a good feel for how does it know to make this? How does it know where to put it? The answer is probably going to be that they have a lot of proteins and small peptides that also sort of force these things and tell the chitin where to position, where to go. But they don't know that yet. Nevertheless, this is becoming a big interest for physicists to be able to design new materials. All right, so this is the last example of a type of material I'm going to show you, okay? So as scientists, we're always trying to make things, as you've seen, much smaller with really incredible properties. Well, we all, I still like magnets. And as a kid, I was always interested in trying to force magnets to go together, and you'd feel that force and they'd pull apart. Well, those are pretty big magnets. Well, bacteria can make tiny nanometer-sized magnets with a precision that cannot be replicated all that easily by science. So these are bacteria. Before we saw a diatom, then we saw beetles. Now we've seen bacteria. And these bacteria, so if you look kind of like this hazy part is an image of the bacteria. And in here, you see these little black squares are the magnets. And this bacteria has the ability to make these magnets almost always with the same size and to orient them into their cell. So how can they do this, and where do they live? These magnetic bacteria live in the ocean. They're in coastal areas, so if you went swimming in the ocean in certain parts, you might run into them. They're also found very deep in the ocean. Now, a lot of bacteria are motile, meaning they can move around. And they have this ability because they have a whip, and sometimes they have whips all over their body. So normal bacteria move around kind of in this helter-skelter way until they find a nutrient that they like, and then they zone in, and it's kind of a superhighway toward that nutrient. But that's a three-dimensional search. But they think that magnetic bacteria have this magnetic core because it turns out that there are these magnetic lines in the earth. And these bacteria, their magnets are strong enough to feel those fields. And what they think is instead of randomly running all around to find nutrients, that these guys are sort of like on a superhighway on this magnetic field, and they're able to navigate on that magnetic field because of this magnetism. They just keep trucking until they find the right environment to grow. So let's look at some of those magnets. So that's up here. These are just really cool pictures of magnets out of bacteria. So this is one organism that makes this sort of square magnet. You can see every time it makes a magnet, it makes it exactly the same. And that is where bacteria beat scientists every time. Small and reproducible. This guy, I don't know if you can see him, kind of makes these needle-shaped magnets. And he aligns them, the fat part down, the needle up, and every time he makes them exactly the same. And so on and so forth. Sometimes round crystals, sometimes oblong crystals. These are the mineral magnetite almost always, sometimes another type of mineral crystal. They're free of internal defects. Again, something that bacteria can do, scientists have a hard time with. Anywhere from 35 to 120 nanometers. Very small size distribution, right? This one is about the same size as that one, which is the same size as that one. And again, every different species has its own type of magnet that it makes. The most important thing is they're not wasteful. Bacteria use the least amount of material to achieve the greatest amount of magnetism of any type of synthesis of magnets anywhere. They are the best at making magnets. They are the greenest at making magnets. That is bacteria, all right. So magnets are formed also, just like photonic materials, just like the silica glass houses. These are made by genetic control. It's the genes that tell them how to make this material. So how do they do it? Well, it's not dissimilar to how diatoms are made. The first thing is you got to make a reaction vessel. Okay, so in this case, that's sort of shown here. You take that oily membrane, and it sort of comes inside and makes a balloon. All right, and that's shown here. So you got to make a reaction vessel. That's called a magnetosome. The second thing is you have to get iron that's out in the ocean into that cell. Now, that's not as easy as it sounds, because some environments of the ocean, they don't have very much iron. And some environments do have a lot of iron, but you still have to get it inside of the cell. And the cells are kind of tricky. They only like certain things to come and go. Well, bacteria are very sneaky thieves and very clever. And what they are able to do are they can make inside of their cell, they make these little things called siderophores, which are little tiny molecules. And they make these inside their cell, and they pump them outside of the bacteria. And these molecules grab iron from everywhere they can find it, even from other organisms, and they steal it. And they bring it back into the inside of the bacteria. And that's the same thing that happens in your body when you have a bacterial infection. Your body tries to actually pull iron away from those bacteria, but the bacteria, smarter, and they have these abilities to grab iron. And so that's how they can survive. So you got to get iron. First you make a bubble, then you got to get iron, then you have to make a magnetic crystal. Well, it turns out that when the cell decides to start making magnets, it tells itself to also make particular proteins, okay? And these proteins are directed to this oily bubble. And when that bubble gets formed, those proteins are now transported into that bubble, but they're facing the opposite way than they were earlier. And so to make these magnets of defined size is sort of balancing by the vesicle size, or the bubble size, as well as the types of proteins that are in there, and the balance of those proteins with all kinds of other factors. And the bacteria has the genetic information to know how big this should be, effectively. But this is pretty amazing, because the bacteria have to control when to make this bubble. They have to know when to make the protein and what protein. They have to control the amount of iron, because it turns out that iron's a nutrient, and you need a lot of iron to make these magnets. But if you get too much iron in there, it's really toxic to itself. So it has to really control how much it gets in there. And they control the amount of oxygen and the amount of pH, too. And then because it's magnetic, these magnets align inside the bacteria, and there are a few other proteins that actually help it align. All right. Whoop. There we go. So just to wrap up on the materials, I hope that what you can appreciate from these different examples, the diatom, the magnets, and this optical material, is that nature makes materials with incredible precision. These are beautiful structures. They're incredibly strong for their weight. They make materials in minutes to hours. What I didn't tell you about that diatom is when it starts to make that one side of that glass house, it does that in 15 minutes. So it makes it in 15 minutes. So by the time you're probably done making, maybe 1 hour, it's done making its entire frustral with that incredible precision that you see here. So not a lot of time. They don't have to characterize these materials like scientists do. And the reason that they don't have to characterize them is two things. One is that it's very reproducible. They're very, very good materials chemists, these organisms. So because it's programmed from the DNA to the materials, it's always reproducible. And the second is that the reality is if they've made a mistake and that diatom dies, that diatom is gone, right? So they don't actually end up having to characterize the materials all that well. But I hope you can see sort of the beauty in nature, not just the beauty that we see in the macros, in the world that we see with different trees and flowers and things like that, but that there's a large beauty in materials that nature makes at this very, very small scale. So I'm going to take the last little bit because I want to tell you about what scientists do, if this makes any sense. So I hope that this will help you. So, like I said before, when I was in high school, I didn't have a really good feel for what scientists did. I was good at math and I took science, and my mom would say, oh, you should be an engineer or a scientist. I was like, oh, I don't want to do that. And I didn't really have a feel for what the scientists did because I thought that physics was looking at waves moving in a tank and a pendulum because that's all I learned. And I thought chemistry was orbitals and all these other things, and because that's all I learned. I didn't really know what scientists actually did. And so let's go back to some of the examples we saw earlier today. So what you saw is that we're very interested in understanding what things look like, right? So a lot of those images are from electron micrographs and microscopy. So we need a whole set of people, different types of people, that are able to understand how light or electrons interact with materials. And then they understand that part because they're physicists or physical chemists. And then together with engineers, they build these really complicated instruments. And this is all part of science as well. Without this, we can't see those amazing structures that I showed you before. Then you have a whole set of people that discover these cool bacteria, right? So it was an ecologist or a biologist, first in the 60s from Italy, and then in the 70s from Woods Hole, who figured out that there are these bacteria that have magnets in it, and they didn't know why they had magnets. They didn't know what they looked like. They just knew they had magnets. So it takes a whole field of people who like to go out and look at what nature has. Then it takes a whole group of people, biochemists who like to look at proteins, and molecular biologists who like DNA, and inorganic chemists who like iron and silica, and material scientists who then characterize what types of magnets they're making. And this is a whole group of people who look at these types of things. And then you have theorists and modelers. All they do is work on computers. All right? They don't mix chemicals. They don't design instruments. They work on computers. But they have a really important function because they're able to explain to us how come this structure looks this way and gives us this color. So my final example here, because I haven't told you anything about organic chemistry, and that's a field of chemistry that might interest a lot of people, is this. Long ago, there were natural healers and a lot of other people who knew that this tree called the Pacific yew tree had a natural ability to cure diseases. They didn't know what was in this yew tree. It just went by lore for a very long time that this tree had natural remedies. Same in New Mexico. There's a lot of different natural remedies that we have. Well, so it took people like natural healers and anthropologists and biologists to discover these really cool trees that have this natural ability. Then there's a whole field of science called natural product chemistry, natural product biology. And they then took this information, they took the leaves of this yew, or the evergreen part, and they crushed it up, and they separated each part of that mixture, that gamush, and they figured out which one cures cancer. And they said, oh, fraction 12 cures cancer. Let's figure out what fraction 12 looks like. And those natural product chemists figured out that this molecule called Taxol, which was one of our best defenses and treatments for cancer, looks like this. And that's what a natural product chemist does. But you don't want to just use all your natural resources and constantly grind up the Pacific yew tree to cure cancer. So there's a whole group of chemists called synthetic organic chemists whose job it is to figure out how to make this synthetically. So they figured out from this part to this part to this part to this part. Finally, they figured out how to make Taxol. So if you like to build things and you like to understand how come the structures look the way they look, you might be a physical chemist or a physicist, all right, or an engineer. If you like to walk around in the field and find unusual organisms, you probably are an ecologist or a biologist. If you like to figure out how come nature knows how to make these funky materials, you're probably one of these guys in here. That's where I am too. And if you like to make materials or synthesize Taxol and anti-cancer drugs, you're probably an organic synthetic chemist. And I was one of these long ago, a natural product chemist as well. So I hope that you can understand a little bit more about what scientists do and some of the cool materials that we had here. And I'll just give you my email address. If you have a question about this, you can also email me, and I'm happy to answer questions.
Jennifer, thank you very much.
Yeah, thanks. Thanks.
So we have time for a few questions. If you have any, don't be shy at all. Whatever you're going to ask.
Yeah.
When you're talking about making the diatoms, and you're talking about a particular kind of compound, silicon, maybe, and creating sort of a bowl
Yeah.
with the silica formed around.
Right.
Does that mean that every diatom uses a different
Yeah.
molding compound?
Yeah, so that's a great question. So let's see if this answers it. It looks like each diatom makes its own type. Each diatom has some kind of polyamine, some kind of silicon, or mixtures of a lot of different ones, some types of silicidines, etc. But every diatom has a different silicon, all right? So it's kind of like, every grocery store has a type of Jell-O, and every grocery store is only allowed to have grape, cherry, or lime Jell-O. And when you mix lime Jell-O with this one, it makes this pattern. And when you mix, grape Jell-O or whatever with polyamine, it makes that pattern.
I have one.
Uh-huh.
So for the magnetic organisms, you showed the lines of magnets. They're lined up partly by magnetic forces,
Right.
I would assume.
Right.
So these are little dipoles that are lined up?
That's
like
right.
that, like that?
Yeah, so these are permanent magnets. So each one is, as I understand it, is a crystal itself. And they're permanent magnets, and they're aligned. Their dipoles are aligned as well, which is pretty amazing. I mean, if you wanted to make these, that's a non-trivial thing. It's really, really difficult to make magnets like this, and nature does it.
Other questions?
What would you postulate the magnetic stuff in the bacteria is good for?
Yeah, so it's navigation. They navigate on the magnetic lines of the Earth, and basically they move along. It's kind of like that's their superhighway, are those magnetic fields, and they navigate along that superhighway based on magnetism. And what happens is, these bacteria are funny. They actually don't like a lot of oxygen. And so what they do is they navigate along that superhighway until they find an area that doesn't have very much oxygen. And then they decide to flourish and grow. And so it's purely for navigation.
Could you find the same bacteria, like not on that magnetic highway?
Yep, that's a good question. And I think the answer is sometimes you do find them.
What was the question?
Oh, so sorry. The question was, could you find them other places than this magnetic superhighway, basically? And my impression is that you can. But the question is why, I guess. Why would you find them anywhere else? Because they've got this magnet. You'd think it would hold them on that superhighway. I'm gonna say I think that you do, but it's a darn good question. A great place to look for information about this kind of stuff is, I don't know, the website called How Stuff Works. You guys probably know that website better than I do. How Stuff Works, and also Wikipedia. And those are great ways to find out how these things, or answer your own questions too. Yeah?
So are the magnets strong enough that you could suck a bunch out of a bottle of water?
Yeah. Yeah, so this is really cool. So when I was in grad school, I didn't know that much about these magnetotactic bacteria. And someone gave me a little tiny tube of the bacteria. And when you don't have the magnet around and you sort of shake them up, you can just see that the bacteria are all dispersed. But you just bring a tiny household magnet, a tiny one that doesn't even have a very strong magnetic field, anywhere close to that, and all of the bacteria slunk onto the other side of that wall of the tube. So even a tiny magnet that you have at your house will move those bacteria. So people are kind of interested in being able to manipulate that and use it for other types of molecular biology.
They're doing anything with it now?
Yeah, so, well, one is that people are using these same concepts that they've learned from these magnetotactic bacteria to make small magnets. So we are getting really good at making magnets. Kannan Krishnan's group in Washington, he makes these little tiny magnets, and they use the same sort of principles that these bacteria use. What a lot of people, I think, would like to be able to do is to actually have regular bacteria that have these magnets in them so that cell biologists can use them to sort of purify everything out by just putting the magnet on them and pulling the bacteria one direction. So, but we can also take these silifins out of the bacteria and get them to make magnets for us on the lab.
Thank you very much.
Well, thanks. Thanks for your questions.
So many of you, this is the last cafe we're having this spring. The next one will be next fall.